Okay, I guess we are almost ready. People are still coming. It's my pleasure to introduce my very good colleague and friend, Professor Beth Pruitt. Beth is probably, I would say, is one of the closest colleagues and friends to me in the community. And it's a privilege that she's here. She's visiting our campus to talk about some of the recent initiatives in the Advanced Center that ABIS just recently started, and many other activities here. Just a brief bio about Beth. Beth received her bachelor's and master's degree from MIT and PhD from Stanford. She worked on piezoresistive cantilevers for characterizing thin film gold electrical contacts during her PhD. And she's probably all of us in the MEMS community, we all know each other, and each one of us is known for something. And I would say Beth is probably known for her expertise in piezoresistive piezo devices and the wonderful work that she has done in this area. And now she's bringing the whole area of the biology, which I think is truly exciting. Beth is the recipient of many awards, including NSF Career Award. And just this year, she received the 2010 Denise Denton Emerging Leader Award, and I'm personally very proud of her. So with no further ado, Beth Pruitt.
Thank you, Reza. And thank you all for coming. I know this is one of the busiest weeks of the quarter, or semester, I guess, for you guys, right? So appreciate you taking time out this afternoon for me to tell you a little bit about our work on MEMS, but I guess more so, I'll tell you a little bit about the sort of a Microsystems themed seminar, I understand. So I'll tell you a little bit about what Reza says I'm known for, piezoresistivity, near and dear to my heart. So for those of you who aren't familiar with it, I'll give you a little overview of that. But more importantly, I want to tell you about the applications. And so not just piezoresistors for piezoresistors' sake, although that's a fun thing too. So what we're interested in for our applications of these piezoresistive sensors, which are force sensors ultimately, is the study of mechanobiology. And for good or bad, when you're looking at various mechanisms of mechanotransduction, or how biology transfers these signals, mechanical signals, into downstream signaling cascades, it's everywhere, right? So it's in remodeling of bone, it's our sense of touch, right, our sense of hearing, even our pulmonary system, right, is stretch-activated ion channels. Cardiovascular system certainly has a lot of force generation, but also force sensing and pressure sensing. So there's actually quite a bit, which leads to just so many things in the candy store to consider for mechanobiology. But I'm going to tell you about a couple that we're really focused in on in particular. So in terms of those opportunities, again, these are very broad as well, but we are interested in cell reprogramming. So what happens to cell signaling in terms of differentiation pathways, in terms of how a cell chooses to not just divide, but to divide into what, or differentiate, or de-differentiate even, between cell type signaling mechanisms. So part of that de-differentiation or differentiation is partly due to signaling. It could be cell-to-cell, or it could be paracrines, it could be factors. And we're very interested in how these things come together in a sort of ensemble for tissue engineering purposes. So we aren't necessarily trying to engineer a heart patch, but we're trying to enable the engineering of a heart patch for cardiovascular health. And the other thing that we're very interested in is more of in terms of a dynamic mechanobiology, and I'll explain what I mean by that in a minute, so mechanobiology versus biomechanics. But some of the challenges of this particular project, up top it's a myocyte, because don't know what a heart cell looks like in culture that's been isolated from a mouse, eating on the scale of about seconds, and it turns out, and I'll show you some images of a stem cell derived cardiac myocyte later, they look nothing like that. And they don't have the morphology, they do beat, but they're not coordinated, they don't really talk to each other, and so we would like to get from here to there. And then down here, there's a wonderful organism called C. elegans, which is a nematode, which is touch sensitive, has a pre-programmed behavioral response. So here we're watching my colleague, Miriam Goodman, poke this guy with presumably an agar hair, although it could be a calibrated fiber polymer suture. And what I mean by calibrated is they take and buckle it on a microbalance. And then they call that a 1 micronewton probe. And that's the smallest force that they can sort of semi-reliably calibrate. As a nice protocol from Marty Chalfie says, the other ones just blow around in the lab environment, right? And you have no idea what force you're applying. For the mechanical engineers in the audience, when I say the word buckling, you realize you also have no idea what force you're applying. or particularly when it buckles. So in terms of the time scale, this is really just for scoring, did it respond or not, is kind of how they treat it in that community. But we would like to preserve the same ideas that they get from the simplicity of an eyebrow hair assay with a MEMS sensing force cantilever assay. So that's the other project I'll tell you about. Now, you'll notice that those two videos are very different time scales. So the worm's ion channels open at least in a millisecond, maybe faster as far as we know, and the behavioral response happens faster than a second. So on the left, I'm showing you, again, that sort of manual simulation. On the right, this is where we're at now, which is this little cantilever centered under the objective, and then we have an XY stage that can move the worm underneath the cantilever. I'll say a bit more about that. That means we can apply and record the real-time force profile and also the video response indicator. The nice thing about piezoresistors and why we settled on them, we weren't, even though I do like piezoresistive cantilevers, we weren't necessarily married to the idea of that. I'll show you some other things that we do in our labs. So we use electrostatics, we do use polymer sensors. But they cover a really large range of force with a single dynamic range, meaning their resolution and their maximum force can cover, say, 3 orders of magnitude, which is something you can't do with these other methods. So atomic force microscopy, yeah, you can cover a large range with it, but you have to change the cantilever for each of these bands to get there. Also, the photodetector in an AFM is not so fast, so we really can't get to kilohertz speeds with that. So we had to sort of, think about what was the best way to access this force displacement map, and then also access the bandwidth of needed for our assays, and then comparing what are the sort of range of applications people have used these other assays, optical traps and magnetic tweezers, etc. Where does C. elegans fit in? And depending if you're talking about the individual neurons, because we're interested in those in culture, as well as in the whole worm, we're looking at much larger forces and displacements than previous, and yet at the same kind of compliance, the same stiffness as single cells. So those are kind of our challenges. The good news is that if you are careful about your design, that you can actually create a probe, not the same probe, again, they're different for design rules, but that will probe a worm or can stretch a molecule. So we now have sort of optimized these cantilever force sensors. A student working on sort of next generation cantilevers to go out to 100 kilohertz with real-time force recording, because essentially once you get above this kilohertz range, there's no way to actuate that with any off-strip mechanical actuator. So I won't talk about that too much today, but by putting a piezoelectric on board, we can now record and actuate at 100 kilohertz. Very small distances, but that's for things like studying the sense of hearing. So a nanometer is a lot in that case. So more about this worm. It has about 300 neurons in his nervous system, about 1,000 cells in the entire, I shouldn't say he, it's a she, it's a hermaphrodite. Well, we have worked with some males, so they're very rare. Males are very rare in nature. I should call them she's because they do, or I don't know, hermaphrodites, whatever the verb is for a shim, can self-fertilize. The nice thing about that means that once you have a mutation in that genome, it will propagate it to the progeny, and very quickly. So each worm has about 300 progeny, so you have lots of worms on a 3-day cycle. They reproduce. They live about 3 weeks. We study them at a particular stage development called L4, larval stage 4, before they've developed actually the gonads and the reproductive capability. So that just decouples us from the mechanics of the reproductive system, since we're interested in the mechanics of that. So in terms of the body plan, if we chop through them, yes, there is some other stuff here in the center of this, but the things we're interested in are in this kind of shell, set of shells. So the cuticle, touch receptor neurons lie right underneath the cuticle. There's 6 of them, the gentle touch neurons that we're interested in, TRNs, which you'll see what's called. And then these body wall muscles that allow the worm to locomote. And of course, this whole locomotion behavior circuit depends on body wall muscles, depends on the touch neurons, and so there's a signaling that I'll show you. But essentially, what we think is happening, a little more detail on this, but is that this touch receptor neuron actually has mechanically gated ion channels. And so the proteins that are in this ion channel have been identified. These proteins are found only in this ion channel. This is a cross-section now to blow up through the neuron. They have 15 protofilament microtubules. Well, that doesn't sound so interesting, except that basically the only place we have 15 protofilament microtubules and the worm is in mechanosensory neurons, like our hair cells. So this is a kind of interesting thing. The mechanics of these microtubules are probably playing a role. And then on the outside of this, zoom in a little further with the ion channel. And so this is all putative, right? We think there are some links that are transferring force. And so what we do essentially is, we kind of, I refer to it as, whack-a-worm. So we try to have more quantitative whack-a-worm so we can figure out how is it that we open this ion channel. And the open probability, it turns out, is proportional to pressure. So it's not just an on-off, but the probability of seeing this opening depends on it's normalized here to pressure, and the reason that Miriam's lab discovered this was because when they applied the same force to different probes, they got different profiles of a probability of open. And then they said, oh, actually we use different diameters of probes, let's go back and normalize it. Ah, so it appears to be pressure sensitive. And that kind of makes sense, I'll tell you why in a second, but this is a cross-section of, actually it's an embryo. So there, 4 of them exist in the embryonic stage, so we can dissociate the neurons from embryos and then start to look at them in culture. We're doing that now. And then this is a GFP image of the neurons in the body. They look huge here, huge being relative. I didn't mention the worm is like 50 microns in diameter and a millimeter long. The neuron body is about 2 microns in diameter. Neuron process, which runs about half the length of the worm body, is 200 nanometers in diameter and has about 30 of those microtubules in its cross-section. So if all these things are mechanically mediated, we need some fine scale tools to figure out what's playing what role and whether anything's open. So behavior at the largest scale, this is kind of like a network diagram, if you will, that's used in literature. So these anterior lateral microtubule neurons, that's the ALM, ventral, ADM, and PLM. So these are, and then the posterior is the P. So then these innervate the motor neurons and sort of stimulate, so kind of contract muscles on either side, right? This is what leads to this sinusoidal behavior. People have done laser ablation studies. People have followed cell lineage from first cell to single cell, or single cell division, all the way through fully developing worms. That was done by John Sulston by watching loads and loads of these embryos develop under a microscope. So a lot is known about what role these things play. And again, sort of either a fiber probe or an eyebrow here, we can kind of, can we play with this worm all day? Yes and no. They actually do eventually habituate to the behavior, and they'll stop playing that game. But there's also another assay that Miriam's lab does, which is basically to first pop the worm to relieve the pressure so that when you go and make an incision near the neuron, you don't have the neuron come spilling out. But then they go in with a recording pipette, a patch clamp pipette, and they actually grab on with a 1 micron patch pipette to the 2 micron cell body in the worm, and the worm is glued down to its head and its tail. And then what we're looking at here, sorry the contrast isn't great, but that's a video with an outline of a probe, just a glass probe. And again, they've sort of tried to calibrate at least bending of this probe against how much force they think this might be related to. But then when we actuate the probe, oops, okay, animation's going crazy there. Let's see. Oh, that movie's not playing. Well, we can imagine the probe hitting the worm. But when you see that, you actually see a lot of compliance. And so you actually see the whole side body of the worm move in this case because you're kind of doing it from the side. They can't actually do it from the top and figure out anything about the force they apply because you can't see the probe. And it all depends on the deflection of the probe. So in order to do this, you have to do it in the focal plane. The nice thing about piezo cantilevers, you can imagine, is that you can do them in any orientation. And then we can start to query, and we will soon, spatial and orientation top side along the worm. But what I wanted to show you is actually the voltage drives the actuator, and we get a voltage which you can sort of infer a force from. And then we get this, if we're recording potential, so we're in the patch pipette, we see a depolarization of the cell. And we see it on the onset of force and the offset of force. And then we see this flow of ion currents, right? And there is, if you do these things too close together, yes, you can't, get this. But what do we have in our fingers? Something about 300 Mechanian corpuscles that exhibit a very different morphology, but the same response. And so this is an ongoing and an off-going response. And so it's kind of interesting to see this off-going response. So what's causing a Mechanian corpuscle is thought to be energy storage. So it's a neuron that's come out to your fingertip, goes all the way back to your spine. And this is basically what allows you to kind of habituate to long time contacts, but it kind of notes, when you're sitting on a chair, it tells you when you make the transition, and when you stand up, it tells you to make the transition. So it's a rapidly activating neuron. And it's thought that these little bundles, this kind of shell structure, is very compliant, so that when I squeeze it this way, I squish, and I get immediate force transfer because it's viscoelastic, and I compress the neuron and it responds. But then all this stuff relaxes because it's very soft and fluid, so with viscoelastic creep, it unloads, and then it's thought when I take off the force, that of course I've stored energy like a spring, and now it compresses the neuron again. That's the hypothesis. The worms do not have a Mechanian corpuscle. They have a touch of the neuron, like I showed you, and this is just looking at the cross-section of it. We think the microtubules could be playing that energy storage role. So this is just an SEM reconstruction from Dr. Cueva in the Goodman lab. So he took serial constructions, high pressure freezing, sliced the worms into 15 nanometer sections, put them through a TEM, and then reconstruct where the neurons go, how many, how big. And yeah, it's pretty cool. And I'll do the reconstruction from this paper in a second, but essentially, this is with the cell membrane without, but it's also connected by an unknown protein. I refer to it as spectrin-like. It's a very compliant, filamentous connecting protein. We don't actually know what that protein is yet. Juan's working very hard to identify it, but this could act as an energy storage mechanism, so basically the compression and outgoing, and then transfer the force to the ion channels. We don't yet have an idea of exactly how the force is transferred to the ion channels. Until a couple of years ago, this was a prevailing model, so basically I've got an anchor on the ion channel in the ECM, sort of underlying the cuticle, and I've got another anchor that connects it to the cytoskeleton, and so that when there's external loading, I get some kind of shear pulling to something that then opens the ion channel pore and allows ions to flow. That was kind of what was the prevailing model, and the proteins were even semi-identified. Well, MEC2 is going to go to the protein VEC712, or what is some length microtubules, and MEC5 is out here. And so Juan started this EM reconstruction not expecting to find this, and then had to do a lot more of them when he didn't find the prevailing model. But it turned out that he used gold immunostaining, so these little gold 5nm beads with antibodies. So you basically throw gold antibodies, or gold antibody tag labels, and then see where they sat, where they tagged, right? And wash them up. So it turns out that the ends of the microtubules, a little bit hard to see here, but is in red. You guys kind of make out. And then he doesn't plot every microtubule. He just starts plotting the microtubules from this cut, which microtubules start at that section, and then track them from the cut to their termination. Some interesting things came out of this. So it turns out that microtubules begin their life, where they kind of generate spontaneously, because it's not one microtubule that goes from one end to the other end of the hundreds of microns long process. It's many. There's average 30 in the cross section. They're kind of tortuous path, etc. But they start kind of near the center, and they slowly spiral out to the side, towards so their terminus, the end point, is always near the membrane. In which case, if their closest point to the membrane is the terminus, that's where I should see the anchor. So he looked at the end point, looked for the anchor, and found MEC2 nowhere near. So the gold is actually labels of where he looked for the MEC2, and then you see the endpoints. It's not, for the size of that protein, these are like on average 50 nanometers away or more from each other, and maybe 200 in some cases. Not enough for this protein to span that distance. So this got a lot of pushback for them, but a lot of interesting things came out of it, including we have to rethink how is the energy being transferred to the ion channel. So now this idea of the spectrin network connecting the microtubule to the membrane and stretch is something that we really want to test. The other thing that was interesting is that we found that, or something Juan found, that the ion channels are, we already knew they were punctate, and they knew already from immunolabeling, immunohistochemistry, that they were approximately every 2 microns or so along the cell process. He further found that they are in a helical pattern wrapped around, so they're not just on the closest side of the cuticle, they're actually all the way around the touch receptor neuron, so they potentially have anchors into that entire extracellular structure, not just sort of apical surface closest to where the force transfer would want to happen. So that was interesting to us. They're also not all active, so you don't necessarily, they're emergent, they can come in and out of the membrane. So this leads to interesting studies that we hope to do in culture, but one of the first studies that we want to query is this idea of the adaptation mechanism. So in terms of, we saw the ongoing response and we saw the outgoing response, like the Jane corpuscle. How does it recapitulate that? So how do you end up getting a mechanoreceptor current that does this? 2 possibilities. One is what Miriam refers to as chanical adaptation. So is the channel just running out of juice, if you will, or there's some other thing that is causing habituation and signaling, or is it mechanical adaptation? So is it just compliant, so that it just moves away, right? Put a force on it, it just keeps creeping away. So we can test this if we can very carefully and quantitatively fix the force and the displacement, and then watch the case of whether it's, as I hold the displacement fixed, do I have to, in order to maintain the force, do I need to do anything? Or do I need to keep increasing the force in order to maintain that displacement? So in order to test that difference, we need a very good way to quantify the force and displacement. So I managed to tell you a lot about a worm and nothing about MEMS. So what does that tell us about the MEMS? So this was our design criteria. So this kind of led to, Miriam and I meeting, which is we need someone to apply a force with these kinds of specifications. We didn't know these exactly with specification. Very small forces to very small worms. Can you do it? Yes. Okay. Some preliminary experiments later, we figured out this is kind of our target range. We now know the transverse stiffness of a worm, for instance, or the range in which they lie sort of normally. And we kind of came up with, all right, here's the resolution, here's the range, piezoresistors can do it. So that plot that I showed you is kind of a, plotting against everything else. Is it worth the investment to go design a custom system? We thought so. And oh, by the way, can you do this while we're patch clamping under a microscope in a wet environment? Sure. So the nice thing again is you can bring this little probe to the patch clamp setup. And we're now doing that, so we're replicating this whole force clamp and bringing it to Miriam's lab on her patch clamp microscope. And so in a month, maybe, actually, as I'm talking, they could be doing the measurements. We're now set up, at least, and they're doing the calibrations. So we make the little cantilever, cartoon of how they work, apply a force at the end, it bends the little cantilever. We put a strain gauge at the root, so that's where we put the piezoresistor. The strain profile of an ideal beam looks something like that. And then take the output of this resistor, which changes its value with strain, put it into a Wheatstone bridge, signal condition it. We get some kind of relationship between force and stiffness, so we can calibrate the displacement sensitivity exquisitely. We have to know the stiffness pretty good. Right? I would say at 10% we probably can calibrate the stiffness pretty well. And then there's a bunch of things, knobs we can turn on the piezoresistor, but the doping concentration is never perfectly at the surface, so we're never just sampling the surface stress. We're actually getting involved with that stress profile. We're getting involved with the dopant profile, which here we're showing an idealized step profile, not the case for a lot of what we do. And here's the expected noise. So if we want to get a measurement down, and this knee, by the way, for most piezoresistors in the literature is about 1 kilohertz. We've done a lot of work to be able to push that knee, in some of our cases, down to 0.01 hertz. Because if I want to measure out of hertz, right, if I want to measure creep, I better not have a cantilever that's drifting faster than the thing that they don't want to measure, right? And so I need the noise to really suppress the noise in this region. So that was a big design criteria for us. So in order to do that, we have to understand that a little bit better. For those who aren't familiar with piezoresistivity, it's a strain gauge. Smith in 1954 was at Bell Labs, and he discovered that in silicon and germanium, that there was an extraordinarily high gauge factor in these materials. And it has to do with their single crystal nature, the change in their band gap as I applied strain actually changed the conductivity and changed the mobility in the material, which is a little bit different. So here's kind of the strain gauge formula. If you used a strain gauge before, it's a response ratio, there's a strain. It's largely, in our metal strain gauge, it's largely a geometric effect, right? So I change the length in the cross-section, and thus I change the resistance with strain. There's an additional effect called piezoresistivity, which is that change in conductivity, change in resistivity. And this is low, but accounts for the difference between a platinum strain gauge and a nickel strain gauge and an aluminum strain gauge. So they actually, all metals do have a piezoresistivity to some degree. It's very small. Most strain gauges have a factor between 2 and 4. Silicon strain gauges, so he made these little bars of silicon. He also found it was anisotropic, so he put current or voltage and then measured the change of the strain by having these bars and measured. So we actually, I won't blame Smith for this notation. It actually comes from the Curies with piezoelectricity, but we end up with a 4-subscript notation, right? Which is what direction is the current, what direction is the voltage, and then the strain has 2 dimensions, x or y. It's the normal of shear. So we have these highly convoluted coefficients that we can design with. So very cool. Thank you, Dr. Smith. And then thank you actually to Lord Kelvin and his disciple Tomlinson. So Kelvin first noted piezoresistivity in 1856. Convinced his student Tomlinson to go off and spend years measuring or change this minor change in resistivity of metals. And he plotted them for all these different metals for us. And so we know that it's also not linear, it's not a fixed value, it changes the strain. And then conversely, someone went back and plotted Smith's values, Conda in 1982, and came up with a theory, at least to match, and I think he has a theory that matches the N-type pretty well. And then you can see this is a polar plot, so showing that depending on the orientation, I can either get a lot of piezoresistivity or none. And so, being careful about the orientation in your design.
What's the size of the current you can measure now?
I'm sorry?
How large current you can measure?
The current.
How big a current?
Oh, did Smith measure?
No, you.
Do we measure? Oh, so we try to use really small currents so that we don't have heating effects. Maybe even a milliamp would be generous, but if we try to stay around maybe a milliwatt in power is kind of more constrained since it's a heating.
And how small are the dimensions of the devices you can use this for currently?
So we have the smallest ones where we now have a paper we're putting together on the thermal design once it becomes a thermal limit. But these cantilevers I'm going to tell you about, thermal is not the problem. The tip is far enough away from the heating, so we can operate them at 10 milliwatts.
You must distribute the devices of the size, so how small can you make individual?
How small, in theory, when you look at the limit, how small could you practically operate them?
How Are we talking sub-millimeter, micro?
Oh yeah. So the dimensions might be like micron type dimensions for the width of the piezoresistor. The smallest cantilevers are 10 microns width and 300 nanometers thickness. Those are those super fast ones I told you about. Those ones, the heating is a brutal problem. I'm not going to show you those data today, but those we have to design very carefully to keep from basically boiling the sample, right? The tip would get really hot, so we have to make sure that we don't.
Yeah, I went through you. I think you went through this neat design, but what is the actual speed you can operate with?
So in our case, we're limited by the off-chip actuator, and that is about 1 kilohertz. It's for a Polytech nano positioning system. For the ones that have a piezoelectric film on them, we have made them separately with piezoelectric films and made them with piezoresistors, and the new process we have them together, so can't claim that that one is working yet, but the piezoelectric alone out to 100 kilohertz.
Okay.
Yeah.
Thank you.
And those ones are those are a little bigger. But basically, you need to design the natural frequency 2 or 3 times higher than the speed you want to actuate at, so you stay away from bringing. Okay. There'll be a quiz on this equation later.
Anytime.
Actually, a quiz on the colors. Anyone notice the color differences?
Yes, I noticed the color.
Okay, so blue is basically things, parameters you can set by geometry, and red are the parameters you can set by process. And I guess what I want to point out here mainly is that these are the 2 dominant sources of noise. There are many others, but this is just in the piezoresistor, not the whole system. And then our ability to tune the sensitivity. So we can maximize the heck out of the sensitivity, but if we don't minimize the noise, we don't really get anywhere. And it took us a long time, as obvious as that sounds, to figure that out, right? So we were constantly trying to maximize sensitivity and sort of paying attention to the noise, and we had this equation, but we weren't really thinking about it until a couple years ago. One of my students says, "Why are we making the cantilevers quieter than our amplifiers?" This is just ridiculous, right? And we're losing sensitivity, so this equation is, losing battle. So we now just kind of look to all of the weakest links in the system, and we optimize the cantilever noise only to the best noise in our system, and then we turn up the juice on the sensitivity. But the geometry is highly convolved, right? So we kind of you win, you lose, and then the process is actually as well. So basically, there's no such thing as a free lunch. We can't get low noise and high sensitivity. You have to trade off between the two.
What's noise for you?
What's that?
What is noise for you?
What is noise? I'll show you a typical noise plot, but this equation here really depends on the alpha, which is the quality factor parameter, and I kind of jokingly call it the almighty alpha because we don't really understand it, and some people attribute it to surface effects and some to both. Alpha for us might vary from 10 to the minus 3 if we're making something in polysilicon, which we seldom do, down to 10 to the minus 7. And so basically getting that number smaller gets us lower noise. And then we might have, basically we have cantilevers that are down to sort of, down to thermal noise, so like KBT.
So you are, when you say noise, you mean thermal?
I mean thermal noise, and I mean 1/f noise. So this is a Hugo noise, so that's the frequency of that noise. Yeah. Right, so this is the 1/f. This is the 1/f.
Do you know how to handle 1/f noise?
I'm sorry?
Do you know how to handle 1/f noise?
Well, we're trying to reduce it. And depending on the source of the 1/f noise, you can modulate and demodulate, which is how we get the data, but you can't demodulate your modulate your...
Because otherwise it will be a very long discussion.
Yes. You can't modulate and demodulate your way out of 1/f noise that's intrinsic to the sensor. But we do modulate and demodulate our way out of all the other 1/f noise from the amplifier, so if that's what you're asking. So that's how we actually get these data, because these are quieter than the amplifier, and so in order to measure the noise, we actually have to do this modulation demodulation trick where we do everything at 300 hertz and then deconvolve it back down. So that's Ali, is a circuit designer in our lab.
So I'm assuming, we'll talk about that.
Okay. Essentially what that means is if you can get a decent model for your thermal noise, for your 1/f noise sources, and so these are the dominant ones for us, and we've looked at a lot of other ones, including, like I said, the amplifier noise, the mechanical noise, the thermal-mechanical noise coupling, and these are still our 2 dominant ones. And for the range we want to operate frequency, the 1/f is the real dominant. But essentially, Sungjin put this plot together, and the 3 of these guys in my lab, Joey, Sungjin, and he have coded this into a MATLAB optimizer. So if you have your constraints, can't go above this power, can't go above this voltage, or if you had found your constraints, you can't go above this doping, can't go above this size.
How do you model physics within MATLAB?
No, the models are built into, you actually have to have an equation, you have to have a model that you put in, and then have MATLAB optimize on multiple models at once. So you go through a parametric optimization, trading off, so you have to seed it with a guess, and then you can do this for multiple guesses. We're coming up with a local optimum. You can't say it's the optimum. So we end up with a local optimum, but if you do that, you can find that by trading off between design parameters for cantilever thickness from 0.5 to 25 microns, that's what the color code means, that we can actually capture, this is 10 to the minus 12 to 10 to the minus 3, I think. So we're actually capturing 9 orders of magnitude in that force axis, and then similarly on the displacement axis, so kind of piconewtons to micronewtons. Not again with the same cantilever, but that's the operating space of a single cantilever design. That's operating space of, within the thickness that you might get. So where can we access in the design space? So that's what allows us to go from molecules down here to worms up here.
What's the light gap between the two?
Oh, he's basically showing just those are the the boundaries.
Oh, okay, so it's a boundary of like a three-dimensional shape.
Yeah. So it's just kind of an optimization plot of kind of here's the workspace, the design space that you can optimize within. The bound limits for the constraints he gave it, which was primarily to say you can only go from 1/2 to 25.
The cantilever is fixed cross-section?
No, cantilever is not fixed cross section. The only thing that we constrained is that we told that you can operate between 1/2 to 25. So this is just a plot to show the design space. It's not.
So I mean, if you move across the cantilever that way,
This is not a plot of a single cantilever. It's a plot of the design space you can access in force and displacement.
yeah, I'm looking at the back picture.
Right? What do you want to know about that one?
If I move away from the support,
Yeah. So this particular optimization is for a diving board geometry, but we also have a multi-leg option, so we can look at multiple legs. But those cross sections, given our fabrication constraints, are still rectangular cross section.
so if it is a rectangular cross-section, if I move away from the support, is it fixed or it changes? What does it change if it changes?
It is the same in this particular simulation and in most of our devices, although it can be multi-leg. You can tailor that though, and so again, if you have a
But
tapered
because I
model,
think you're going to get advantage if you change it.
you absolutely are. So that's another discussion. One of my colleagues is optimizing piezoelectric energy scavengers, and he finds rectangular cantilevers are absolutely the worst shape for that.
Exactly.
In our case, and for the simplicity of the beam theory, and also to make sure that we stay linear within all the bounds.
There is beam theory that we have here from Antman and others that we can handle any kind of varying cross-section you want.
I mean, many people can handle varying cross-section. In our case, for our optimization for our needs, we can stay within all of our models and all of our linearity and get where we want to go for the rectangular cross-section. So essentially what happens once we've made this, and that's just a small piece of the system, is we have our force sensor that in this case we can go nanonewton to micronewton. It's 2 different cantilevers to cover this range though. And then this is, as I showed you before, the worm underneath the microscope. That's, what does it take to get this image? So the cantilever is part of it. We put a glass bead at the end to have a constant cross-sectional area, contact area, regardless of the orientation we want to go to. And then we can change the size of the bead, obviously, but we skeletonize, we can track, and we can position the worm underneath here. So it's all on a microscope. It can either be, in our case, in our lab, we have it mostly on a stereo microscope, a dissecting microscope, with or without fluorescence, and then we can track and do this. In Miriam's lab, we're now putting this onto her patch clamp, which has both an upright and inverted, and you can bring the patch clamp in next to this, and then do the patch clamping of the worm. So you can either have a freely moving worm for the behavioral assay I'm showing you here, or you can have a glued down worm. So I'll show you the behavior assay, which is essentially, we do some post-processing imaging. There's a new student in the group who already taken a look at this and said, oh my god, the computational overhead, and of course you have to do this all offline. So he's already figured out a scheme for doing this quickly with like single frame images, and then being able to just go find the centroid quickly and do this real time. So he's wonderful. So essentially this is a plot. This is we took this idea from Arabi Samuel's lab, where they do this for swimming and locomotion. This is a plot of the ventral and dorsal curvature of the worm. And so if it's just moving along in a sine wave, then we see this straight line essentially of going from ventral to dorsal as the worm moves along over time. And then you get an idea of the gait. Get an idea of the gait time, right? Just in terms of the time scale here. So once we skeletonize it, then we can use this for the tracking. We go back after doing some of these, and we can look for subtleties in these gaits. One thing we learned is that here's a worm just freely moving. That's a control. And then we go and do the same thing at 100 nanonewtons and 1 micronewton. And what we're looking at is sort of, it's when the numbers appear in the top up here is when the force is being, or up here in the right corner is where when the force is being applied. And then you can score the response by eye, but what we found is that this is going to lead to errors because there's actually, at the lowest forces, you don't see it here at 1 micronewton, but at 100 nanonewtons and below 1 micronewton, they're in a wet environment. They're calling on wet agar. And so the surface tension and the capillary forces lead to ringing when you unload. So of course the worm responds to that. So how do you differentiate? So we go back into video imaging, and we then score them. So we put the desired profile here, We score them for that window. We don't remove the cantilever. Of course, the worm's moving underneath it, so the force is changing ever so subtly, right? Because he's kind of underneath the cantilever for that 100 milliseconds. Then they dwell for 2 seconds at a 0 force. So they try to use the feedback control to stay at 0 force without unloading. And then over here we remove, and you get the ringing. So we can score the behavior response here, and you'll see that we see, when we first started looking at these, we're like, wow, they respond to 100 nanonewtons. They're responding to this, to the pull-off force, which is actually a micronewton. And so once we went back and did this kind of finer filtering, we realized that actually a micronewton, they'll respond to 100 nanonewtons, appears to be about their threshold, so they don't respond most of the time. And in touch response is a percentage game, so 10 times you touch them on the head and tail, 8 out of 10 times they respond, 80% touch threshold is kind of how they quantify that. So what this allows us to do is not just look at loss of function, but also to look at even gain of function. So do you actually get hypersensitivity, which is a particular symptom of some diseases that today could not be scored. So you actually get a more response with force, particularly in the ion channel. So now, in terms of filling out this curve, this was the data that had been published before using these microvine phi hairs, they call them the little buckling fibers. And then if we expand it out to our data, we find that it actually follows the typical probability open curve, right? Of like we'd expect 50% of the time it will feel it at about 100 nanonewtons, about half the time they respond, about 20% of the time they respond at lower forces than that. So what does this mean to us in terms of back to the microtubule and the adaptation story? And we still aren't there yet, I'll admit, but we're getting closer. So what this means is that if we know when they respond and we know what force we apply to get them to respond, we really want to go in and do those patch clamp assays with the ion channel. So we're gearing up to do that because then we can record the patch clamp response with the force profile. We can see, again, is there a lag, is there a latency, is there gain of function, loss of function. But in order to understand what kind of displacements that were causing, we kind of needed to know at least some gross mechanical model of the worm. So when I deform a worm by 1 micronewton, does that mean I deformed it by 1 micron, 10 microns, 50 microns? It's only 50 microns thick, right? So we hope not. So one of the first things we did actually were to figure out what is the spring constant of a worm, if you will. And so here is wild type data. Spring constant of a worm is on the order of 1, like from 0.6 to 0.8. I think you want to look at the median or the mean. And that it's biology, so large inherent variation. We have verified that the inherent variation of our system is much smaller, and we indent glass before and after every experiment, make sure everything is still good. But here's a wild type worm that's been glued down with the experiment, head and tail. Kind of hard to see, but there's a little comma-shaped worm there, DPY5. That's a genetic protein that encodes the cuticle structure development, and that stands for dumpy. So the worm community is very clear in their names of genes. The fly community is not. They have strange names like pickpocket and things. So if this worm is dumpy in the worm community, we're going to call him dumpy. This one is long and skinny, so we're going to call him Lon. There's another one that's called small, SMA, right? And he's actually the same aspect ratio, but smaller. So there's a lot in the worm community that's been defined. So this wild, dumpy 5 worms will give more dumpy 5 worms. That's genetic mutation, right? A whole plate of dumpy worms. So the prevailing model that I don't think I mentioned, but essentially it had that shell and it has the stuff inside. Prevailing model until a few years ago was that it's a pressurized hydrostatic skeleton. So like our tongue doesn't have, it has muscles we control it with, it doesn't have a skeleton, but we can still, locomote our tongue pretty well. The worm was thought to be the same thing, and structurally it kind of is, but the skin of our tongue is thought not to give much structural integrity to the tongue. Same thing was thought for the worms. What we found, we were kind of surprised that actually this ultra-structured shell, which we kind of define as the cuticle, the body muscles, that outer area that I showed you, actually does seem to contribute some structure to the worm. And in terms of identifying what it is, we're still working on which component is the cuticle, which component is the muscle, etc. But to test the hypothesis, we expected pressure to matter, so the ultimate experiment, of course, is to pop the worm and measure it before and after. And yes, pressure matters, but you notice that it still maintains quite a bit of its initial stiffness before and after. So that's pairwise, pop the same worm. And then population studies, again, so head, center, tail. And then cuticle mutants, the dumpy 5, if he's really blown up because of a ratio of the pressure is really changing, I would expect that if pressure is dominating his stiffness and then his cuticle has been deformed, but the cuticle doesn't matter, just the shape of pressure matter, then if it's kind of like a balloon, right, and it's now short and fat, he's got a higher pressure, right? So I've blown him up to a higher pressure. Same thing with the long and skinny one. So by the prevailing wisdom, if the cuticle doesn't matter, long and skinny, it's because you should have a lower pressure, a lower stiffness if the pressure matters. And we found just the opposite, actually. So the W's are stiffer, which sort of suggests that they're getting some rigidity from this, being their aspect ratio, so this ultrastructure.
So is it a single in-kind model, or is actually pieces of identical sections that they're connected, that they provide many of the motion and many of the stiffness, like in larger models of snakes and things like that, where you actually take simple mechanical structures?
No, we basically took, we looked at the literature for models of pressurized bacteria, of pressurized viruses, and hair cells. So things that have an internal trigger pressure, as it's called, and also have a shell. And essentially what led us to this hypothesis was that when you indent this, it should, if it's just kind of like indenting, Jello, I should get what's known as a Hertzian behavior. So a sphere indenting into a flat, I should get this kind of exponential behavior. It shouldn't be linear. And we saw linear behavior. And we tested all the different substrates, tested hard, soft, all kinds of things underneath the worms, see if that mattered. And it was definitely in the worm, that it was intrinsic in the worm, it was linear. And a shell model, when you indent and locally buckle a shell like that, that leads to that same kind of behavior. So it's an approximation, very much so. But essentially says that we've got some kind of ultrastructure that behaves more like a shell than like a solid.
What happens when you puncture dumpy and long?
So we didn't puncture dumpy and long. That's a good experiment to do. But actually something that we became sort of more interested in, I'll tell you in a second, but and what we did do in this other case though was to look at for wild type, and the other reason we didn't do it is that there may be other aspects. What's that? We blew them up, so we didn't pop them, but we did blow up the wild type. There may be other mutations in dumpy and long, so even though we say we know it's a mutation in dumpy that leads to this development, it's never a clean mutation, and so people don't really know what other mutations are going on. So there may be other things. So popping them would be reasonable, and we still don't know for instance, we need SEMs to know if their cuticles are the same thickness, but to do that requires a pretty intense couple of months of someone high pressure freezing, slicing into EMing worms, and we just haven't committed to that yet. So instead what we did was we cultured worms in hypertonic solutions, and we essentially blew them up. And so there was a paper that had come out the year before that was very nice talking about osmolarity responsive worms. So they can actually express water very quickly when you put them into a saltier than usual plate. And so then you kind of you transfer them in a less salty and sort of blow them up. So we looked at the transfer between those, and we actually saw, well, you do see a shift that's not statistically significant. And so again, while pressure matters, it doesn't appear to be the dominant factor. So but this gave us, overall, like I said, some confidence that, when we're indenting, we have some idea of a kind of gross global modulus, or a spring constant of the worm is more appropriate. I'm not quite sure how one gets a modulus of a worm, right? Because there's a very complex, heterogeneous, non-anisotropic structure.
So
But
if
it's,
I take this model you have over here, I slice it.
this model here.
Yeah, and I slice it, and I think of what I was referring to as a cell before, not what one means a cell.
This is not a cell. This is a 1,000-cell organism.
Let me finish the question. So what I referred before is the cell is not what biology means cell. I mean the cell is.
You mean a unit cell in a finite element sense?
I said that's right. I slice it like this, and I handle it non-uniformly by looking at together to get a better model of what behavior should I have with these individual pieces, so I can get all the motion and everything like that. Isn't that a better model than just trying to do a?
Well, we aren't modeling locomotion. So Arabi Samuel's group is trying to do this for locomotion, and they're looking at various models to fit that.
Right.
Even they're using a viscoelastic rod type of model, which it's really hard to account for the active and passive components, etc. In our case, we're trying to look at what governs the transverse stiffness, and to figure out what is an appropriate force displacement relationship. So when we apply 1 micron into the worm, how much that means that we're displacing it, 1 micrometer. So we really didn't have any idea of, when I squish a worm, how much deformation does how much force generate. So this gives us a gross idea of force versus displacement was really where.
Yeah, but you want to put the all together in this elongated model, right?
No, actually that wouldn't help us for the touch response. We really need to know local deformation. What we're really trying to get to is what's going to open that ion channel. How is force transferred from the cuticle, through the cuticle, to the touch slipper neuron? How are the microtubules? What kind of loads are they feeling? So what we really need, and we're going there next, is the transverse model of the worm is not necessarily going to help us. The transverse model of the touch receptor neurons with the microtubules is where we want to go next. So now we're moving towards dissociated touch receptor neurons, looking at knocking out the microtubules, depolymerizing microtubules, and then getting a better model of how they handle force is actually going to help us a lot more to understand this. So that's kind of where we're headed for the next element. So I wanted to just reorient you to this. These are the muscles, and then again, sort of the cuticle structure, there's neurons inside. So again, when I sort of refer to the shell of the cuticle, the muscles, etc., we realize that muscle activation is playing quite a role in maintaining this body stiffness.
Is the muscle only on the inside?
It is in quadrants, as shown. Yeah.
What I'm asking is that it's only dorsal ventral, or is it?
So it actually crawls. I mean, they crawl on their side, if you will.
So
Right?
it's-
It is, yes.
So it's side to side, but they don't have, is that like, they can't go both directions?
No.
They can't go circular?
No, no.
So they can only go one plane?
Right. And they have more in their head for foraging, so they can lift up their head.
But your body's going to use
That,
Yeah.
so,
All right, so basically I want to show you this again because what we started testing most recently is this idea that the body wall muscles are actually maybe the dominant shell modulating behavior. So we take your worm, here's your normal drugs, 2 different worms, levamisole puts them in rigor, and this BDM, and we've recently moved to another drug that $10 for a quad. Mucimol. And I know the reviewers want more experiments with Mucimol. Sorry. Anyways. So the BDM and the limits all let us test the hypothesis of like kind of relaxing and stiffening muscles, and does that change body stiffness? So Brian and Pierre spent a lot of time looking at how this changes body stiffness. Luckily, we happened across from Alan Skotchak's lab. He shared with us an optogenetic strain that has channel rhodopsin and tail rhodopsin in the muscle. So blue light and yellow light is much cheaper than mucimol. So we can basically flash the worms and we watch them shrink when their body wall muscles contract, right? And then you can put the yellow light on and they relax back to the original length. So we can look at changes in segment lengths, free segment. Again, this is, we can't get it perfect. We have to try to figure it out from where they're free to move. So it's not perfect, but it at least gives us an idea of whether or not is stiffness changing, pre, post excitation, and during excitation, and does it return to the same value? So it mostly returns to the same value statistically, and it definitely, what we found is it gets stiffer. So stiffness is modulated by body muscles. I don't think I have the data here to show you, but it turns out that their touch sensitivity of these worms relative to baseline is also decreased with stiffness, which is awesome because that fits the hypothesis too, right? That basically the body stiffness, or this spring constant, if you will, or of the worm is modulating its force response. All this is important because this is all in the feedback circuit again, right, for worms to respond to one another. So again, body wall muscles. We also looked at a couple of different mutants, uncoordinated mutants, uncoordinated. We looked at 2 different ones. One where the muscles aren't attached to the body wall, and we also tried doing this with RNA silencing. So it's really easy to feed worms bacteria that express RNAi, and then the worms will have that RNA signaling silenced. So we tried to do this by detaching. It's a really sad-looking worm. It doesn't fully detach the muscles from the cuticles. You can't quite look at the effect of what we want to do with channel rhodopsin, detach the muscles, and then simulate with light, watch the muscles contract, but not be able to pull the cuticle with it. But it does all kinds of great things to worms. So not good. But we can look at things like spacing. We can also look at collagen, GFP expressing lines. We can watch the furrows. They have these little furrows on them. So we can watch the furrow contraction on the drug. You can't, again, do the GFP while you're doing the channel rhodopsin. There's all these technological things. But we can look at and see that indeed the interfurrow spacing matters. I didn't mention this, I'm not going to get into too much, but think of the worms, especially at the adult stage, like an accordion. So if they're actually contracting, it turns out they have these little furrows in their cuticle, and it may be, we can't separate out this hypothesis yet, so muscle attachments, and then there's these little spaces. Maybe that as the worm contracts its muscles, that part of that effect that we're seeing in the stiffening is due to the muscle being activated and stiffer, but it may very well be that it's actively stiffening the cuticle because it's collapsing the accordion. So that was why we wanted to detach the muscles from the cuticle to test, which is the right model for that. And then, as I mentioned, so Joey has been working on extending these for the worm studies to single neuron studies, but also hair cells with Tony Ricci's lab. And so for these, again, we need much faster biome times, much smaller tip deflections, and much higher bandwidths. But what we found is that for these, at least theoretically, we should be able to operate competitive to optical trap for doing this, but with the control of having a cantilever that you can go in and place and pull against any surface. So for that, Joe went to, again, this role comparing aluminum nitride and PZT films. He found that actually, in the design trade-offs for these actuators, even though PZT can give you larger deflections and larger displacements, larger coefficients, that the aluminum nitride, by the benefits of both fabrication and its match to the silicon, is actually superior for our application. So he's sticking with this. And then for him, it really matters that you not get the cantilevers too hot, because they're really small and they're going to be in solution with the hair cells. We don't want to have a thermal effect in there. So he's trying to minimize the heating at the base of the cantilever, but designing to control the temperature at the end of the cantilever. So here he has just a 10-degree temperature rise, or 5. Here it's unacceptable, right? That's like a 15-degree temperature rise. And so that's a constraint that we have to add recently to the design. And so his are really small. Someone who'd asked about dimensions. So here's a 15 micron scale bar. This is actually these devices. These ones are a micron thick. And then these are not tested yet, but they have a piezoelectric and a piezoresistive loop, so you can actuate on board. We also use our cantilevers. They have nice force feedback. We use them as sensors. We use them to calibrate everything else around the lab too, so calibrated pretty well. We've been using these EMS force posts to do measurements of force generation in cells, and then calibrate how good, and compared to silicon cantilevers, the post stiffness actually varies quite a bit across the wafer, probably acceptable, but at least it's good to know in our opinion. So calibrating these little EMS things, because we wanted to use these, other people have used them too, but we wanted to measure the force generation of those cardiac myocytes that I showed you earlier. So some other methods include culturing them on cantilevers, trying to put them on posts and optically detect that. But again, if you don't know the material properties of the post really well, it's hard to know what forces they generate. And then the cells that we actually are interested in, like I said, are these undifferentiated stem cell, or differentiated stem cells that we made myocytes out of stem cells. So they're not large, they don't look like myocytes, and they're really hard to get to stick to things and to handle. So we had to come up with a way to take these cells, which are 10 times smaller than an adult myocyte, and do the same kind of measurement with them. So we couldn't grab them, we couldn't put them on posts, etc. But Beth's figured out this way to basically do a sacrificial process. So she puts P9chem, a thermally sensitive polymer, that fills around the post, so the cells don't fall down in between them. Some of them, preferentially from the surface treatment, will put their focal adhesion attachments across two posts. Some of them will do something weird, and we ignored those. And then we change the temperature by 5 degrees and flow water through, and it will dissolve the P9chem. And you're left with suspended cells, which is kind of cool. And they do kind of, around them, if you look at the focal, they do kind of, they're not just on the top, they're making an attachment all the way around the tops of the edge. But it was kind of nice because she was able to make the first measurements of, this is an embryonic derived, so this is a neonatal, and this was an embryonic derived stem cell human. And actually, interesting, this neonatal and this is actually from Lonza. So if anyone has bought cells from Lonza, these have been through the works, right? They've been dissociated, frozen down, shipped to our lab, and then thawed out, and they still beat. But we don't, I wouldn't call that a monosystem, or with a test technology. But they do generate around the same amount of force, which for your reference is about 10x smaller than an adult myocyte produces. And so again, if we want to get to, the point of you having your doctor tell you someday, oh, gee, you had a heart attack, but we can repair that, and we're going to take these embryonic derived stem cell myocytes and put them in your heart. Wouldn't you like them to generate as much force as your original myocytes? It's kind of our philosophy. So we wanted to create the gold standard for, physicians to be able to tell their stem cell therapies are going to work or not. Yeah.
So you have these two posts on the cell. How do you measure the force?
Oh, so you're watching. Oh, there we go. You're watching a displacement.
Oh, the cell is being watching the displacement of the post.
And then, so from having calibrated them in the last slide, we then, these are rather tall in order to get these small forces. These like an 80 micron tall post. A lot of people use these little 5 micron other things that aren't beams. So try to a lot of people also like to use this technique called traction force microscopy. So put beads in a substrate, put the cells down on top of that, and watch the beads move. So very similar, but you have a continuum of substrate. And again, it's very hard to calibrate that. If you don't know your modulus, all bets are off as to what is going to come out of that. So we were trying to come up with a way to calibrate that, and Gaddard and Chelsea put together this this idea of a traction force tool. So we basically touch down with a little preload sensor so that you know when you hit the surface. And then at the surface, you shear between these 2 feet. And again, you get kind of a force displacement, which ultimately is what you want. You want how much force the cell generator needs that far. So you shear, watch the micron, you know how much force it took to do that. And then you're kind of calibrating your traction force shells. So it works okay, but it actually is a pretty tedious undertaking as well. So useful, but we also need ways to do things on more ensemble or large scale populations because biology has amazing variability and we need to watch it do different things over and over again until we converge on a mean of what they really want to do. So, and then this last thing, it's a short section just to tell you what we're up to on sort of polymer dynamic cell culture. So how do we kind of torture cells on a mass scale basis and why? I already mentioned the ideas, you know, our collaborators in the Cardiovascular Institute are very interested in creating a heart patch. We're very interested in making them, make sure it's functional, doesn't use animal products, if they can avoid having to use animal cells, and when they get to animal cells in use, so it probably has to be not your own cells to induce pluripotent cells, probably has to be, most of us don't have embryonic stem cells anymore, unless someone saved your blood for you. So maybe mesenchymal stem cells, that's a possibility, but probably you're not going to mail order cells from Lonza from a rat in your patient. So we kind of need to come up with a way to vary some of these different parameters that we know affect cell culture and help them, you know, test whether or not their cells are effective. So this is part of a collaboration that involves a biomaterial engineer who's working on tunable substrates, tunable stiffness, induce topography in them, and tune the amount of biochemical ligands in there. So she creates engineered peptides that mimic laminin binding, a little bit carbonectin binding, so it has the user or RGD sequences in there. And they can tailor how much they put in there. They also have neural cadherins, and they're working now on VE cadherin, which would be cardiovascular cell adhesion molecule. And we're really interested in using their materials and putting them into these mechanical environments, both to test and to strain the cells to see how they respond. And sort of conventional culture with myocytes, you usually have to electrically paste them to make them beat, and so to coordinate them and cause that cascade. Ultimately, it wouldn't be really like just one pacemaker cell. So current cultures are done by putting electrode across a bath of cells and pasting them all. It's not how your heart works, right? There's actually a pacemaker node, and it propagates, and the cells communicate that. So we really like to move towards having one stimulus cell that you actually feel. And this is a different set of collaborators. So they've developed a myocyte, an embryonic cell-derived myocyte, that has channel rhodopsin in, so they can pace with light. So then the idea would be to pattern your pacemaker node in the corner, and then have the other cells be cells that are going to propagate and watch that. So in terms of looking at how do we do high-throughput cell cultures, a little space, it's actually not been, I mean, I'm sort of surprised it's not been done that much, right? Just a few people are making these like stretchable cell culture systems, so look at what happens. There's a commercially available system called the Flex Cell, and it's a 6-well plate, single pressure, you have to do all your strains at once, and there's sort of a limit on how you can pattern the bottoms of those. Luke Lee had these very nice parallel culture systems where you put load with a little C-trap to catch the cells while they're flowing until those stop in here, and then could cross-perfuse with different components to different, there's a gradient generator, put different gradients of chemical factors on them. So very nice, you can use it with a microscope, but if you actually want to use these next to do some kind of tissue engineering with, this is a throwaway chip, there's no retrieval, so it's great for fundamental studies. It's glass, you can't strain cells, so, you know, we had some nice conversations with him about wouldn't it be nice if we could stretch this chip and how would we do that. So the Flex Cell is great, but it's kind of, this is it, this is large, the whole thing has to go on your incubator, and this is amazingly, anyone here has affiliation with Flex Cell, I apologize, it's a Windows 95 machine. They've not updated their software. I have to sell you a Windows 95 PC if you buy their system. So we like that idea, but we want to be able to strain multiple things. So we created just a little control board. It's like this big with a little programmable controller, solenoid valves that will modulate lab vacuum. So you also don't need to have an external pump in your lab to do this. It's not noisy. And then you have a little plenum down here that draws the vacuum on a deformable membrane in each well. It has cells in it. It gets stretched. This is how it's made. It's pretty simple PDMS. Acrylic. And then how it works is essentially each membrane is stretched across a different size post. Larger post when I suck down on the membrane underneath. Less of it gets sucked down, so there's less strain on top of the post. Smaller post, more of it gets sucked down. There's a limit on how small that post can be before the strain fields are nonlinear over the post. We're mostly interested in cells over the post because that's where the strand field is biaxial and uniform. So there's also been a lot of proposals for inflated balloons, but you can imagine if you inflate cells on the external of a balloon. If we blew air into this instead, all the cells are seeing a different strain, right? So it's falling off from the center to the edge. So we really wanted to have the same strain within the central region of the pus. Calibration, care, but okay, calibrate. We can do an axial, simulate, see that it kind of matches, get that nice calibration and simulation. We can also do 3D, so the strains are not so uniform, but we can find, we call it Stonehenge, and we can fill it with a hydrogel with the cells. Do the same thing for protein patterning. You can use Stonehenge and then confine a droplet of like laminin or surface treatment. And then after you've incubated a device with that, you can now confine the cells to the center. So we're going back now to do the experiments better, I would say, constraining the cells to the region of known strain. So our first experiments did not constrain them, and cells are seeing the non-linear strain, or the highly non-biaxial strain at the edges. Strange, interesting things happen. Here's one experiment I'll show you from this, though, which is that we cultured, this is a maiden Darby canine kidney epithelial cells. They want to form a confluence sheet. Turns out the way in which you plate them is very important. So you plate them down in low calcium media so that they adhere very well at a known density, change back out to normal calcium where they can form their calcium-dependent cell-cell adhesions. And once they have divided once in that culture condition, they go quiescent if they're confluent. So you don't have any plaguing problems of like how many cells and how many more cells and which ones are coming and going. So you have some normal cell death, but for the most part, the layer is confluent and stable for about a day for most of the things we want to study. So we're interested in how cells adapt their cell-cell contacts under load, so they start straining them. How does that impact their tissue? So it's really hard to see in this on my screen as much, but there's actually 3 colors seen here. There's blue in the nucleus. There's red at the edges, which is actually an E-cadherin. And the blue is DAPI, the green is vinculin, and there's a little spot of red there, so that's a cell-cell contact. So those cadherins are, if you can see this a little clearer, we'll just go to cartoon of what we're looking at. It's a little clearer. We actually have an endogenous label on the cadherin, and then we go back to antibody on the vinculin in that image, and antibody on the nucleus. But our collaborator, the Nelson Lab, has GFP labels and RFP labels on a plethora of proteins associated with cell-cell contacts, so we can start to ask questions about that connection, again, from intracellular to extracellular, and also cell to cell. And again, it's the same protein on both sides, this cadherin-cadherin complex that causes cell-cell adhesion. And what is the role of these different associated proteins, vinculin, alpha, beta, actin? So this trimer of alpha, beta, and cadherin is the thing that in pretty much all systems, right, us, all multi-cell systems, is this cadherin complex, which is cadherin calcium-dependent adhesion molecule. And it was not until this past year, documented or known that it was mechanosensitive. So what's interesting is that the data I'm going to show you, we actually saw before we read any of these papers, and our collaborator said, can't be true, too weird. But then all these other papers came out and said, look, cadherins are mechanosensors. So we told them, when we do these, that's terrible, I apologize. Projector. I don't think projector's not yelling it. Okay, so let me go to the thresholded. Okay, so the thresholded at least, we thresholded all of these, and we looked at different strain levels in one, looked at time intervals, and again, this is not stained because that would be a sacrificial, right? So this is endogenous, so the cells are producing a red label on their cadherin, so they're endogenously transfected and expressing it. So at 2 different densities, and we see this at certain densities. This is confluent, this is super confluent. We've gone 50,000, so it changes, and we need to get more data on, no error bars. But essentially, what we see is that this is vinculin recruitment. So if we mask immunofluorescence to a mask, so we threshold the cadherin and say, where's the cadherin, and then go back and look, where is the vinculin? Vinculin's everywhere in the cytoskeleton. And it turns out under load that the vinculin is actually recruited to the cell-cell contacts and reinforces them in some way yet to be determined. But that was kind of the point of these papers variously testing this by doing things like giving drugs to the cells to knock out their cytoskeletal or their muscle or their motor proteins, and basically so cells couldn't generate forces. So no one has yet reported that cell-cell contact, when you stretch them externally and don't do anything else to disrupt them, so the only way to relieve the force in other studies, as I said, is to drug the cells, which will have other consequences, but it turns out we see it with external load as well, so we see vinculum recruitment coming to the contacts, which I think is super cool. So we'll continue to explore that. Hopefully we'll have plots with error bars and show on that soon. But in summary, I think there's a lot of benefit to miniaturizing and to parallelizing some of these assays, both for worms and for cells. And I would like to thank the numerous people. No, my lab is not that big. That's my lab. This is our cardiovascular tissue engineering retreat with joint program. In fact, just Mariana, you're in there somewhere, right? So this was a retreat that we had with F3 sites who had similar interests on tissue engineering or cell-cell communication. It was a really fun retreat. So had a lot of good collaboration with folks here in Maryland, as well as Wisconsin, and a couple other sites that are interested in these questions as well. So I would thank these folks and our funding sources. I'll take any more questions if you have them.
Okay, we have time for a few questions.
So is it surprising to people that cells have on mechanosense?
It had not been documented before, so people knew about mechanosensation, if you will, between the cell and ECM, the extracellular matrix. But in terms of cells communicating to each other, there had not been identified any proteins or any.
Okay. But it is reasonable, right?
No, it's completely reasonable, but in fact, part of the reason our collaborators were surprised by it was that they didn't put force into their prior studies, but a lot of this stuff is done, on glass or in petri dishes. It's done in static conditions, and so it's very hard to see it unless you know what you're looking for. You're going looking for it, right? So people went looking for it in some of these proteins based on both steered molecular dynamic simulations or based on what people had seen in these, like talent is one that's involved in the focal adhesion and integrant binding. And so people had seen this by pulling on it in proteins purified in culture. When people had looked for, say, vinculin in the role, which is affiliated with other functions in the cell, it's out, everywhere in the cytoskeleton, it does get engaged in focal adhesions. So no, it's in focal adhesions, it's not in cell. It's not brought there, we don't know how it binds in, we don't know what it binds to, we don't think it's force sensitive. So previously when these collaborators had looked for vinculin being associated with the coherent complex, they found no sign of it, let alone it being upregulated under load. So it shouldn't even be there, but they had done it in these static culture systems where they flayed open the cell and then tried to do immunochemistry on the cells where they had kind of treated it having phase cell-cell contacts, and they saw no sign of binding of these other complexes. So this trimer was really thought to be pretty pure. And how it linked to the actin cytoskeleton was not really known. So vinculin being a player in that, and being a player which potentially increases its binding under load, is pretty exciting to the community, as you saw from 5 papers in the past 6 months.
So in the study with the worms, so once you understand the mechanics of how the neurons fire with the tactile application we apply to the worms and everything, and understand sort of the mechanotransduction and all that, then what is that sort of the
Like next week.
Yeah. What are then sort of the human medical applications that benefit from that knowledge?
Right, so
once we can fully understand that, what can we then sort of do with that afterwards?
Right. So the model that I told you about how pituitary and corpuscle works is just a model. It's still not known. There's a really nice review article that came out a couple weeks ago from Alan Lumpkin reviewing what we know about the sense of touch and actually more highlighting what we don't know about the sense of touch. It is the least understood of our senses. And yet it is affected. Neuropathy, if you've heard that term before, is your sort of loss of the touch sensation in peripheral limbs, peripheral neuropathy. It's a side effect of diabetes. So if anyone knows diabetics, and they kind of, they sort of start to lose sensation, and they actually start, if you can't feel that you've twisted your ankle, for instance, then you're going to walk around on it until your foot needs to be amputated. Seriously, is the number one side effect. Well, it sets a pain, so basically any kind of mechanosensor.
Okay.
But if we can understand how they work, then we can start to understand how to restore touch sensitivity. So if the nerve dies, that's one. There are mutants where the neuron dies, and there are mutants where the neuron cannot produce the touch sensation. What's interesting, or not does not produce those proteins. What's interesting is that the worms where they don't produce those proteins, but the neuron is there, actually has a worse adaptive touch response than the worms that don't have the neurons at all. Right. So they're not complete. It's not completely understood because there's other sort of right. There's other accessory. And so one of the points that Ellen Lumpkin studies mouse DRGs. So one of the points she makes in her review paper is that actually it's been very hard to study this in mammals because there's so many sort of backup systems in place. So if this one fails, there's so much that both genetically and in terms of the receptors, like 3 neuropterygical muscles in your hand, that's just one, or finger, and that's just one type of receptor. You have slow acting and rapid acting, many types. But HIV, chemotherapy, diabetes, all result in neuropathy, which could be either death of or failure to function of the nerves all the nerves that give you your touch sensation generate from your spine. I mean, it's different than a worm. They're myelinated. They have a sheath like a coaxial conductor that allows them to. So many diseases are actually a loss of that outer conductor, which is harder to study in worms. But if you can understand signaling of touch sensation, so I should mention that I don't think I've made this point, but something like 80% of the genetic code of worms is conserved with mammals, and their neurotransmitters. They use dopamine. They use serotonin.
Yeah, but the fact that it's conserved is really, that's
Right, but there are or homologs, I guess, is what is thought in a lot of cases. Whether or not this MEC4 protein, the closest thing of the acid sensing channels, ASIC family, whether or not understanding this particular ion channel and how it opens is going to allow us then to rapidly identify an analogous ion channel in a mammalian cell, unclear. But at least in terms of the motivating aspect, the assays and understanding are a lot easier to gain rapidly in a 3-day regeneration system.
So there was one set of slides where you're talking about this cross sections and the mapping out of the cell. In some of those figures, there's kind of a texture around the microtubules. Was that an artist's rendition, or was that part of?
No. Oh, where I showed that
Yeah. You said there was some
spectrin-like protein.
Spectrin-like.
That is actually there.
So it's something. It's a visible.
That is a reconstruction from his imaging data. Yeah. So it is there. There is that network that's.
And you just don't know what that protein.
Do not know what it is.
So you see those attachments, that network.
Right.
So you could go into some sort of finite element analysis.
Knew the properties. Yeah. But it turns out like cross-links between something spectrin-like and the cross-links between something actin-like, and that's one of the proteins that bundles actin, and it's a loose link. It's the one that creates the but the spectrin is the one that makes the sheets, if you will, so the membrane type structure of actin, as opposed to I don't remember what some of the other cross-linking proteins are, but it's actin cross-linking proteins. Essentially, spectrin is kind of what what's thought to give the red blood cells their membrane shape by concave disc, so it gives it structure.
I have a question. Also, for the worm research, they're trying to see what the response of the worm by the macular force, right? Is there any other issues besides the macular force that can change the response of the force of the worm? For example, the shape of the contacting area, because you have
Yes, yeah. So I showed you the data that showed that pressure is actually what matters. So we can apply. This is actually kind of somewhat debate in this kind of touch assay community is what should be the metric by which we compare. And I actually, in general, like we should show all the data, right? But you look at these papers and you might see. Stop the camera here, but some people want to transform everything into a force, and that's actually, and you have to give all the transformation, right? You can't just say, and the force was this. If you don't tell me how you got to it, like in our case, we know the displacement really, really well, and we know the spring constant pretty well. And so we can give you the force, but we're not going to give it to you without telling you how we got it, and that it was displacement. Should we be thinking about these ion channels opening in terms of pressure, force, strain, displacement? That's again why we need a better complete model of this, right? So just sort of saying that something opens at a particular force, if you same force, stiff probe versus soft probe has very different implications. I mean, and so Dennis Disher and other people have shown this repeatedly in the last few years with different cell types, but especially stem cells, that their differentiation depends on the substrate they're on, right? So their phenotype that they want to preferentially differentiate to changes whether they're on a 10 kilopascal gel or a 10 megapascal gel. Yeah, so they,
It might be that they have more capacitors because they've tested on more substrates.
absolutely. And actually, when I started digging into this, this is in the literature back to the 60s, just highlighted. I mean, so, you know, I was like, wow, this first paper is like 2005 or something, people figured this out. And then I found this paper from, you know, 60s, 70s, 80s, people talking about changing the types of gels that they were doing cell culture on when they first figured out, like, Petri dishes and, you know, glass. So, this whole unknown, good question. And, you know, I urge you when you write your papers to report it all. Yeah, but this, I think there's, you know, Amos and I talked about this earlier, this idea of kind of wanting to give up modulus to everything, and I regret that we kind of gave, and we put all the qualifying statements, I've gone back and checked this 100 times in our paper, that, you know, if we take the shell model and we fit it with an effective modulus that lumps together all these things, and we assume it's concentrated in the shell with this kind of thickness, that you just complete ballpark numbers based on other species, other things, and this pressure from the scarus, and then you can get an effective modulus, which, you know, we all know is built on a house of cards as engineers, and it's on the order of this, and all we wanted to do that for was to say, yeah, and it kind of makes sense, it's like a meniscus. Fine, right? And the thing has been cited so, like, everyone, oh, they got E wrong of a worm, their E of the worm, and like, of course, their E is totally wrong too, because their assumptions are entirely wrong is relative. But E is not a material property that I wish we don't use it for anything except for bulk isotropic materials. But that's another story.
Yeah, that's another story. Okay, well, let's thank our speaker again. Students, if you want to talk to Professor Pruitt, she's going to hang